Thank you very much. I hope that in spite of this ugly title, people will be attracted by my talk. But before I start, I would like to thank first Karim Benzerara and Thibaut Corradin, who is not here, but and their team, respective teams, who organized this very nice CNRS school. And it's very surprising to have these CNRS schools because they are as numerous as the fingers of the Milo's Venus. And it's so rare to have this sort of school that let's take the occasion to go on with it. So before I start again, I would just like to present very briefly our team, which is Bio-Geoscience in Dijon. Dijon, you know, the mustard town. The team is divided in four, the UMR is divided in 4 teams. One is Biome, which is a team devoted to macroevolutionary process. And within this team, we have a little sub-team, which is biomineralization, which exists since 2003. And we are 3 permanent members. Let's present them. Until recently, we had Nathalie and Gilles together. Now we have Irina and Françoise. And among the non-permanent people, we had Benjamin Marin, who did his PhD and his postdoc with us. Also Nathalie Leroy and Paula Ramos Silva. And a lot of what I'm going to say in the second half of my talk comes from their data, from their work, also together with a little bit of what Françoise did recently. So I present here a very simplified phylogeny of the metazoans, of the animals, of pluricellular animals, on which I plot the different type of biomineralizations. And in hatched area, you see the skeletal biomineralization, and in full colors, you see the non-skeletal biomineralization. As you can see, many of the biomineralization are of calcium carbonate, of course, not exclusively, since we have also phosphates present in, for example, in brachiopods, and also, of course, in chordates. We've seen that this morning with Philippe Janvier. A little bit of silica present in porifera, in sponges, and also all kinds of other biominerals which are non-skeletal biominerals. And as you know, and it has been said this morning, most of these skeletal tissue or non-skeletal biomineralization were implemented in metazoans at the dawn of the Cambrian times, somewhere in between 545 million years ago and 520 million years ago. Here with calcium carbonates, with these precursor animals, Cludina, and also other animals, calcium phosphate, and again a little bit of silica in porifera. And if you look at the properties of metazoan biominerals, there is one common theme, which is that these biominerals exhibit emergent properties, which means that these minerals are more than the sum of their separate elements. Inorganic phase on one side, which is usually brittle, and organic phase on the other side, which is very soft. And usually combining the two together gives new properties. So an aspect of these emergent properties are found in 2 things. First, metazoan biominerals present a complex hierarchical structures. And secondly, they are organomineral assemblies. I'm going to go very fast on this aspect, and then most of my talk will be devoted to this part. So about the hierarchy in metazoan biominerals, I just remind this very nice picture which was published some years ago by Helmut Kolfen and also Kolfen and Antonietti, who this picture really depicts what can happen on a theoretical point of view, that metazoan biomineralizations are produced via non-classical crystallization pathways. And these, from nucleation clusters, you go to mesocrystals. So adjunction of little elements which just orientate, and this oriented attachment produce then these monocrystal-like crystals. And of course, these pathways are not related to the complexity of the organisms which make these biominerals. Since, of course, we know the system of bone. We know that bone has at least 7 or 8 different structural levels from the single tropocollagen molecule to the complete bone system. But if you look, let's say, between brackets, more simple animals, like here, Corallium rubrum, you see that it has also a very, very structured hierarchy in its structure of skeleton, coming from herringbone units, which are divided in spindles. The spindles themselves are divided in lozenges, which are also, let's say, composed of, again, little fibers and so on and so on. So here you have, again, 7 levels of hierarchy. So this being said, let's go back to the, let's say, organic aspect of the story. Here, in this very simple graph or simple diagram, I just try to summarize the life of a metazoan biomineral, and I take the example of a mollusk. And finally, biomineralization, what is it? Is the integration of different levels, of different components. One part is the development, which means that upstream the mineralization itself, you have a complete array of genes which are involved, which will define, which will tell to the tissue to differentiate and to produce minerals. So beside these aspects, which has been, for example, well covered in the sea urchin model, you have some other aspects. And here, there is what I call the intracellular and membrana trafficking. So what happens within a cell, let's say fluxes of calcium, for example, for calcium carbonate systems, which work together with what I call the extracellular events. And the extracellular events are the mineralizing matrices. And my talk of today is really focused on this aspect. And later on, of course, the animal will die and will just go to the fossil record. So, if this works, yes. Going back to the organic matrix, the organic matrix is has a sort of very simple definition, because when you take these calcified structures, you reduce them in powder, you decalcify the powder, you will extract some organic compounds. And these organic compounds are always a minor part of the whole skeletal tissue, between 0.1 to 4 and 5% in most of the cases. And we know now, after 3 or 4 decades of work, that they are supposed to display a major role in biomineralization. These are what we call our molecular toolbox. And the molecular toolbox is, so this is this matrix. And the concept of organic matrix implies that it is made of a lot of different elements, different macromolecules, which can be proteins and polysaccharides, for example, that are supposed to work together to form the structure, the mineralized structure. But this concept of organic matrix has evolved a lot for decades. And this evolution of the concept of organic matrix goes in parallel to the techniques that were used for analyzing these matrices. And we are going to see together these aspects. Let us remind that the fact that, whoop, I don't know what it means. Let us remind that the fact that the organic matrix is associated to mineralized tissue, in our case calcified tissue, is something very old. Because already in the Middle Age and even in antiquity, people used to extract from bone and from skin a substance, gelatin, And you know it comes from the degradation of collagen. Collagen means which generates glue. So this property was known for already quite a long time. But people had to wait until 1685 before an industrial production of gelatin from bone and from skin. And if you look at also in the very old literature, you had a French guy called de la Sonne in 1751 who made these experiments of calcination of bones. So very interesting to read. Going a little bit further, in the 19th century, the first, and I think this is true, I didn't see other types of analysis before, but the first truly biochemical study of biominerals was made by another guy, French guy, chemist called Frémy, who in a 60-page paper described what the composition of bone, but not only bone, but also octocorals, crustacean carapace, and mollusk shell. And for the little history, Frémy was the guy who invented, in this paper, invented the terminology concholine, which is still now used. If you look at your Little Petit Robert dictionary, you will find concholine. But you don't find biomineralization. Strange enough. Well, this is before the concept of organic matrix. The true concept of organic matrix was set up after the Second World War, especially by people like Rogers, Wilbur, or also Glimcher, on different systems. Roger on bones, Wilbur on mollusks, and Glimcher on vertebrate, bone and teeth. And I like this sentence of Glimcher. The hypothesis proposes that the precise juxtaposition of certain reactive groups in the organic matrix creates highly specific regions which act as sites for the heterogeneous nucleation of the appropriate crystals. So this sentence was written only 7 years after the discovery of the structure of collagen, the triple helix, by Ramachandran in 1953. And already at that time, Glimcher had a sort of quite interesting or quite accurate idea of what could be the role of the organic matrix in mineralization. And we can go further with this different concept by taking the mollusk as a system to work with. Because the mollusk actually sort of synthesized most of the ideas that were, let's say, put forward to explain how the matrix could work. And I represent here in this diagram how people think the system works. Here you have ARCA, and if you make a section here, you will see this piece of mantle together with a piece of shell. So you have the mantle with this epithelium, very thin, which is enclosed by the periostracal layer. And in between, you have the extrusion of inorganic ions together with extrusion of the so-called organic matrix. And by a mysterious self-assembling process, you have the production of this shell. So this is a nice story, of course. And the story started to be unveiled because of the possibility to analyze proteins just by cleaving completely these proteins, so-called amino acid analysis. So this was made in the late 50s. So you can imagine that if you go in the old literature, in a couple of years, 54, 55, 56, many calcium carbonate systems were started to be analyzed by this technique, and especially mollusks, by Belgium team, the team of Grégoire. And at that time, for the mollusk model, what people saw was that the shell matrix was first highly insoluble and very hydrophobic. Hydrophobic means enriched with especially 2 amino acids, residues, glycine and aniline. So the idea that emerged at that time was that the shell matrix should work as a sort of mold around the crystals, something very insoluble in which crystals can grow. Of course, this idea was sort of primitive, but it was the feeling of the period. Then, in the early 70s, some guys, very clever, thought that when you dissolve your calcium carbonate biominerals, you can look not only what goes at the bottom of your pot, but also in solution. And in solution, you have a soluble matrix, which is quite different. So that was the work of Meenakshi and Crenshaw. And just a few years later, Weiner, in a sort of key paper, Weiner and Hood, proposed that, on the basis of amino acid analysis, that the matrix, the soluble matrix, was very polyanionic, negatively charged. So this matrix had something to do with interacting with calcium carbonate crystals. And of course, at that time, in the 70s, but in the 80s and even later, there was a constant problem that people met by analyzing skeletal matrices associated to calcium carbonate, especially for mollusks, but not only. First, about the insoluble part, it's not possible to dissolve it easily. So it's a sort of black box. About the soluble matrix had some certain properties which make it very difficult to fractionate by chromatography. Most of the time, this matrix behave as a smear in different chromatographic systems. So for a long time, maybe 20 years or more, the most efficient technique to tackle this matrix was to do some amino acid analysis, just to break everything, maybe make some fractions by chromatography and then analyze. Okay? So at that stage, people started to think about models. And again, Haddadi and Weiner were very efficient in providing a sort of models where the soluble matrix, the one which is polyanionic, which is supposed to react, to be very active in the interaction with calcium carbonate surfaces, the soluble matrix would act as a nucleator, but also as a crystal shape modifier. And this is these little graphs which depict this. Few years later, Wheeler, an American researcher, found that the same matrix, when you put it into a solution, will just act in a completely different way. It will just inhibit calcium carbonate production. So the test is very easy to do. Maybe some of you have already done it. So you just put calcium chloride in a solution of sodium bicarbonate. In normal condition, you will just record a little change of pH with the production of calcium carbonate. But when you put a matrix, you do not see this effect. So this is called the inhibiting effect. So it means that with these two tools, inhibition on one side, nucleation, we had the yin and yang of biomineralization. And that was, everybody was very happy with that. This also led, in particular, Steve Weiner, but also others, people like Bevelander, Nakahara, to propose for the mollusk system let's say a ternary model where you have chitin. So chitin is a very complex sugar. It's a polymer of N-acetyl glucosamine. It's very insoluble. Chitin would provide a sort of structural support. Then you have the soluble acidic proteins, which are involved in nucleation. Then you have the insoluble hydrophobic proteins. And these proteins would be involved in sort of mold. And later, this mold was transformed into a gel. So that's exactly what I depict here. So in the early 80s, you had this model, which was a sort of sandwich between these three components, beta-chitin, hydrophobic, and acidic, on which crystals grow. This model was transformed 13 years ago into a new model here, where you have, again, chitin. You had acidic proteins, which are making some clusters. And in this cluster, you see, you have this silk fibrin gel. So it was a nice model in a way, but still a ternary model. So does it reflect the reality? Of course not. So in the, let's say, last decade or last 15 years, there were a set of techniques which permitted or which allowed it to go a little bit further in the description of the organic matrix. First, the 2D gel electrophoresis, then the molecular biology approach, and finally the high throughput screening techniques. And I will just go on with these three techniques. About the gel electrophoresis, just a couple of examples. Something that we did some years ago, where you see here the helix. You know, we are in Burgundy, so we like helix very much. And when we can eat them, it's very good for us. And when you, let's say, extract the matrix from the shell of the snail, or the land snail, you see this little amount of spots. Actually, there are many more spots, but you cannot stain them. So only the spots that you can stain are on a 2D gel have this distribution. So you have very acidic spots, but you have also very basic spots. If you take another example, and this is the work performed by my colleague Françoise Imel, who is here at the back. So here you have the Dreissena polymorpha, which is a small muscle. And Dreissena polymorpha, when you extract the matrix of Dreissena, you see also a lot of different spots, from very basic, which are hardly stained, but they exist, to very acidic, which are hardly stained, to very basic one. And you can see also these patterns, which are probably phosphorylations. So we see already with these examples that the situation is much more complex than just a ternary model, right? Going back in 15 years ago or 20 years, 18 years ago, some people had already done a lot of work, especially on the sea urchin model. So this is the team of Fred Wilt and the paper of Killian and Wilt in '96, where they radio-labeled proteins of the skeletal tissue, and then they made this autoradiography, 2 gel electrophoresis with autoradiography, and they could enumerate, in a way, 12 strongly radio-labeled proteins plus 3 dozen of less strongly labeled proteins. So again, here you have a system with something about 50 different proteins associated to calcium carbonate crystals. Then, so if you go one step further, now going to true molecular biology, then I just summarize here what happened in few years between 1996 and 2004, just for the mollusk system. People started to look at proteins using molecular biology techniques, and you see here about 20, I think there is 19 or 20 different proteins which were slowly published year after year. And what do we learn from that? Well, the first protein which was, let's say, discovered was nacrein. It was discovered by a Japanese team, published in September '96. And nacrein, of course, didn't look like what we could expect from the model. Nacrein is a very special protein with a carbonic anhydrase domain, subdomain. Here, carbonic anhydrase is an enzyme, and I will speak later about enzymes. Another carbonic anhydrase subdomain, and in between, a region which has nothing to do with carbonic anhydrase. This is called a GN region, and this region is supposed to interact with calcium carbonate. So that was the first surprise when this was published, because again, something was completely new and did not fit with the sort of simple ideas that we had. Then the second protein was even worse. This is lustrin A. And lustrin A is composed of very long, high number of modules, like cysteine-rich modules, proline-rich modules, which are like pearls on a collar, with a long GS domain, so a domain enriching glycine and serine. Again, some basic domain here, a protease inhibitor domain, so a very complex protein. And this protein, we know now that it's probably sitting in between naked tablets, and it has something to do with the special physical properties of naked tablets. Then there was the mucoperline, which was published also, and mucoperline also is something very unusual in comparison to the previous models, because it has all these tandem repeats units, which are of mucin types. Mucins are a class of proteins. These are the main components of mucus proteins. And we could see for the first time that this mucoperline was really restricted to the nacre layer, so it was a nacre-specific layer. So all these proteins had nothing to do with what we knew from the models. So in short, in summary, what we know from the 2D gel electrophoresis and molecular biology was that organic matrix, especially in mollusk, composed of several proteins, acidic, neutral, basic. Many of these proteins are modular, so one protein is supposed to perform different functions. We could start to put in evidence several different domains, and many of these domains are of this class, which are repetitive low complexity domain, RLCDs. But in the recent years, the knowledge about the skeletal matrices in mollusks started to explode just by using what we call the high throughput screening techniques, which is supposed to allow the analysis of complete protein secretory repertoires. Secretome, skeletome, shellome, so we can use all kinds of terminology to describe the proteins which are associated to mineralized tissue. And this is made basically by proteomics, by transcriptomics, and also by a combination of the two. So I just very briefly explain here what it consists about. On one side, you have proteomics, you just extract your matrix component from the shell, and these components can be further separated or analyzed in bulk. And then they are digested and analyzed by mass spectrometry in tandem. And you generate a lot of peptides, usually small peptides which can ionize easily. On the other side, you have the transcriptomics. So you start from the mantle tissue, the tissue which makes the shell. You extract all these RNAs molecules. RNAs are converted into cDNAs, and then these cDNA are massively sequenced. And when you combine the results of that and the results of that, you finally get your proteins which are associated to your calcified tissue. Said like that is very simple. There are some tricks, but some people are well placed here to speak about that. But whatever the difficulties we have, owing to these two techniques, in few years, people started to massively gain sequence information. And the first person to get this was Karl Heinzmann, a German guy who first got proteomic data from the eggshell, and where he found like 500 different proteins, which are constitutive of the eggshell. He made the same for the sea urchin, and again, a complete set of proteins. And also, he also analyzed other systems. For transcriptomics and for the mollusk model, there was a couple of key publications by Daniel Jackson, so an Australian researcher from the group of Bernie Degnan, who used the Aliotis alzinina, which is a gastropod, a vetigastropod, to generate a lot of transcriptomic data. And the conclusion of these two key papers, which were maybe passed a little bit unnoticed by the community, but for me, to my sense, they are the best paper in the field. These two papers showed that several transcripts, this paper identifies several transcripts which encode secreted mantle proteins of totally unknown function, first point. And then in the second paper, he showed that many of these shell proteins, or so-called shell proteins, have a rapid evolution. So they have sort of, they evolve quite rapidly. So in doing that, Daniel Jackson sort of revealed a part of the unknown, the unknown part of the iceberg. So, summarizing the knowledge, in the recent years, high throughput screening generated an inflation of novel proteins associated to biominerals. And I just plotted here the number of proteins, mollusk shell proteins, that are now known. So 1 protein in 96, 19 in 2004, about 50 in 2008, and now we are more than 300 proteins in 2014. So there is a real dramatic increase of our knowledge, with the expansion of the list of molecular functions which are required for regulating mineral deposition. And now the picture is a little bit blurred. So did we gain clarity? No. But we can maybe go a little bit further on some evolutionary process and also on the molecular mechanisms of biomineralization. So that will be the last part of my talk. And I will take a couple of examples. The first example is the scleractinian coral. So I leave for a while the mollusk model and I jump into the coral model. Corals, scleractinian corals. So they work a little bit like mollusk, I would say. Saying that is sort of approximation, but it's in a way it's very appealing, because the calcification process in corals is an epithelium-driven process. It means that in a coral, the part which is active is the so-called calicoblastic ectoderm, so the ectodermic tissue, which is in contact with the growing crystallite. And things are supposed to happen in this calicoblastic ectoderm. What I presented for mollusk, this small progression of the data and the comprehension of the skeletal matrix proteins, is similar for corals. The first sequence known encoding a shell protein, skeletal protein, was galaxin in 2003. Then we had some partial sequences which were published, and also a couple of new proteins which were published until the paper of Drake and al. In 1.5 years ago, 2 years ago. So now corals are becoming a sort of hot topic in the field. Why? Probably because of OA, of ocean acidification. So now things start to be generated more quickly. So in our case, and that was the work of Paula Ramos Silva, we chose one coral, Acropora millepora, which is a coral, scleractinian colonial coral, which is a part of the big reef barrier in Australian eastern coast. And between brackets, you know that this structure, this biomineralization structure, is the single one to be seen from the space. This is the biggest construction. And this coral was used, and some people in Australia generated the transcriptome data. And on our side, on the side of Paula, she performed the skeletal matrix purification and extraction. And of course, in the case of corals, you have to be very careful, because corals are extremely porous. So if you don't clean them very accurately, you will probably end up with a lot of cellular contaminants. And of course, they completely blur the analysis. So we applied a very extensive bleaching treatment to really get the proteins which are the most associated with the calcified tissue. And by doing that, we ended up with a list of 36 skeletal proteins that we grouped in 5 clusters. About 15 of them have an extracellular matrix signature. Only 6 of them are acidic proteins which are enriched in aspartic acid residues. We have 10 proteins which are fully uncharacterized. 4 of them are enzymes, and we even have 1 which is similar to a toxin. So that was quite new and very surprising. But the most surprising was to come, because among the proteins that we observed, 11 of them had transmembrane domains. What does it mean? It means that, well, why can we say that? First, when you put the sequence in the different tools, you can see that there are transmembrane domains, but there are some signs that indicate us that a certain scenario can happen with these TM domain-containing proteins. Probably these proteins are, they have these transmembrane domains and they are cleaved. And we found in the sequence that there are a lot of potential chymotrypsin cleavage sites. We also found in the set of proteins some enzymes, proteases. And at last, we always found this part, the external part, associated to the calcified tissue, which means that these transmembrane proteins are just, during the calcification, they are just cleaved, and then the outer part, extracellular part, is integrated into the skeleton. So we have here a change of paradigm, that from a system where everything is secreted outside the calcifying tissue and then integrated into the skeletal structure, a part of it is, of course, secreted, but a part is just like pointing out of the epithelium and then cleaved by the enzymes. And here, by having this model, we can imagine that there is real a macromolecular link between the calcifying tissue, the calcifying epithelium, and the end product, which is the skeleton. And Paula did also a lot of analysis just to compare this content of 36 skeletal proteins with other cnidarians which are naked cnidarians which do not produce any skeletal tissue. And she found out that only a part of these proteins are strictly Acropora-like proteins, but some proteins have their homologues in the naked cnidarians, so non-producing skeletal tissue cnidarians, and a lot of them are found in these two non-skeletal forming cnidarians, Nematostella vectensis and Hydra magnipapillata. Which means that if we summarize the knowledge that we gain is that, okay, we first had this set of 36 skeletal proteins, which are grouped in 5 classes. These skeletal matrix proteins are secreted and membrane bound. Only 8 proteins seem to be skeleton-specific proteins. 3 are homologous in Nematostella vectensis, and 9 other proteins are seen in the 2 other cnidarians, which suggest that these proteins, a part of the equipment for calcifying, was actually co-opted secondarily for calcification. But of course, we don't know whether this event was a recent event or whether it was something very old. Well, the results of this study were published about 1 year ago. Now we go to the second example, the example of carbonic anhydrase. So as you may know, carbonic anhydrase is a group or a class or empire, I don't know how to say, but a very complex group of different enzymes. In metazoans, you have only one class or one family, which are the alpha type. And these enzymes, their goal or their function, is to hydrate CO2 to produce bicarbonate. So bicarbonate, it can be considered as the precursor of mineralization, because if you react this with calcium, you will produce calcium carbonate plus a proton. So these enzymes are extremely ubiquitous, that you find them in gills, in lungs, hemocytes, osteoclasts, brain, muscle, and so on, all kinds of tissue. And they are supposed to perform different functions, and probably the initial function was to regulate the intracellular pH. And also they are involved in respiration, in bone resorption, and in calcification. And this enzyme belongs to a very old regulatory system. In metazoans, alpha carbonic anhydrase have various cellular locations. They can be intracellular or in mitochondria. They can be bound to the membrane or attached to the membrane by GPI anchor, or extracellular. Well, we had two models. Sorry, I have to drink. We had two models in Dijon. One is the freshwater bivalve, Uniopictorum. And we could really unambiguously see that the calcium carbonic anhydrase activity was present in the shell matrix. And by proteomics, we found a lot of carbonic anhydrase peptides. And this activity was really dose-dependent, so it was really the true enzyme which was active in the shell. Now, if you take Aliotis tuberculata, a gastropod, a vetigastropod, Nacre's gastropod, we were never able to find carbonic anhydrase activity in the shell matrix. We never found any peptides which suppose that carbonic anhydrase is included in the shell. However, we found two carbonic anhydrases which are mantle-specific by using molecular biology. One is the secreted form, and the one is the transmembrane form. So here, with two mollusk systems, we see that already they work differently. On one case, you have a carbonic anhydrase which is secreted and then integrated into the shell. On the other case, for Aliotis tuberculata, we have a carbonic anhydrase which is secreted. One is here, lying on the membrane. One is secreted but not integrated. So what can make the difference between these two types of carbonic anhydrase? Well, if you now look at, if you make a very thorough sequence analysis going to many different metazoan systems, you will get more than 100 of different carbonic anhydrases. And some of them, they are a little bit different than the others. We found like 33 carbonic anhydrases which possess supernumerary domains. So for example, in the case of Stylophora pistillata, we have carbonic anhydrase domain here, but something in addition here and something here. For Lotia gigantea, the old limpet, giant old limpet, we have a long domain which is supernumerary. For Pinctada, I mentioned before the nacrein, we have this GN repeat domain. For the Strongylocentrotus, we also have, so this is the sea urchin, we also have these domains. And it's very likely that these supernumerary domains are used for integrating the carbonic anhydrase into the skeletal tissue. Well, to make the story of carbonic anhydrase a little bit short, we also, when I say we, this is the work of Nathalie Leroy, plotted all the sequences on a tree. And the tree is actually extremely complex. That's why I do not show it here. But it's in the recently published paper of a couple of months ago. What we can say in short is that about alpha carbonic anhydrases, they have complex evolutionary history, which implies many genes duplication, For example, I was mentioning the giant old limpet. You have you have 17 forms of carbonic anhydrase in this animal. And most of these genes have had completely independent evolution. So in each metazoan lineages, you have independent evolution at different levels, probably at familial levels, at order levels, or maybe at genus level. And we have also the case of these transformed carbonic anhydrase, which have a supernumerary domain. And this is really a phenomenon of gene tinkering. So probably by exon shuffling, these carbonic anhydrase were made and just to perform new function in association with calcification. So I go now to the last example. Okay, I will be in time. The last example is the protein repertoires in mollusk shell microstructures. And this is quite an old story because I just show here some of these old monographies which describe very accurately what is the constitution of the shells. So here there is an example here of a shell of a bivalve, Mercenaria, and where you cut it, when you slice it, you can see already that there are different layers, a very thin inner layer, a middle layer, an outer layer. So in the past, there were people, some naturalist researcher, who really looked very accurately at the composition, the structure of these layers. The first one was Bogild in 1930. Then you had the monography of Taylor and Hall in 2 volumes. And recently, the Carter, for a geologist, it's very recent. So the Carter book of 1990. And in these books, they really describe very accurately how the crystals are organized. And here I show very briefly few of these types. Because imagine that for what we call the prismatic layer, this is, for example, a simple prismatic layer, but you have a lot of different subtypes. The same for nacre layer. You have subnacreous layer. You have nacre in a brick wall, nacre in a columnar nacre for gastropods and also nautilus, cephalopods. You have foliated layer, which is only in calcite, that you see in oysters and in a pecten. You have this very complex thing, which is composite prismatic layer made of very tiny crystals, which are making bundles. Homogeneous layer, cross-lamellar, which is the nightmare for the naturalist, because nobody understands how it works, still now in 2014. So most of these layers are aragonitic. Some of them are calcitic, like foliated, like prismatic, and sometimes. And there was, of course, for 1 century, the question of how are these layers generated? What makes these layers being like this or like that? And so we can sort of go 1 step further. In the case of the Polynesian oyster, Pinctada margaritifera. And this work was performed in the frame of this Adequa consortium, Franco-French consortium, with people from Polynesia, people from Paris, people from Dijon, people from Lyon, and so on. Just to try to, the goal of this consortium was initially to find ways to improve the quality of pearls, right? In some ways, there were some achievements, but as a side project, we also worked on prism, on nacre. So I show here the prismatic layer, which is calcitic, the nacreous layer, which is a well-known brick wall nacre found in many bivalves. So people from Montpellier generated what we call EST libraries that they assembled and found these more than 76,000 unique sequences, very long sequences. And by doing proteomics on our side, and this is mostly the work of Benjamin Marin, proteomics on the prism, on the nacre, we try to solve whether these 2 layers have the same protein repertoire. And the answer is, well, about the Prism matrix, we found a list of 48 different proteins. Most of them are new, were new, not published. Only few of them were already observed or discovered by Japanese colleagues. Interestingly, when you try to summarize the proteins in terms of functional domains, you see that many of them, they have these repetitive low complexity domains, which are different types: hydrophobic, GY rich, hydrophilic polar, like Q rich, acidic, and so on. You had some enzymes, peroxidase, tyrosinase. So tyrosinase is interesting because it's involved in what we call the quinone tanning, so insolubilization of proteins. Chitinase, we had proteins with a signature of extracellular matrix. We had also protease inhibitors. Protease inhibitors are interesting because when proteins are secreted, to avoid their degradation, you need inhibitors. And you had also EF hand, so probably proteins which are involved in binding calcium. So that was what we observed on the Prism matrix. For the Naker now, we have a list which was slightly shorter, with only few proteins which were known before, and many of them were unknown, 32 proteins. Look, on the protein domains, well, smaller variety of protein domains, but still a lot of repetitive low complexity domains, hydrophobic, C-rich, M-rich, and so on. You have proteins which had extracellular matrix signature, also here, something which looks like a protease inhibitor. But if you compare the two protein repertoires together, only 3 proteins are common to the two layers. All the rest is different. So it means that, well, the mollusk, the Penctada margaritifera, it makes its shell, the mantle tissue really secrete 2 sets of proteins which are very, very different. And among the very bizarre proteins, we found this one for this candidate, MRNP34, which was published a few years ago, which was very unusual because it was very enriched in methionine residues. Methionine, this is a sulfur containing amino acids, and this is a very unusual amino acid in biominerals. This is not the one that you find first, usually when you make amino acid analysis. And this protein is, for example, very specific to Naker layer. You do not find it in a prismatic layer. But, of course, its function is totally unknown. So our colleagues from Polynesia made this very nice immunohistology with specific antibodies against one of the proteins, where you really demonstrate accurately that only a part of the mantle epithelium is stained by the antibody, the part which corresponds to the production of the Naker layer, and the part corresponding to the production of prismatic layer is not stained. Similar results were obtained with this one, MRNP34, by in situ hybridization, where you have the nacreous part, the nacreous forming part which is stained, but not the part which is forming the prismatic layer. And again, also we used another technique to also show that there were really some proteins which were specific of the nacreous layer. So the outcome of our study was that the Prism and Naker proteins repertoire are totally different, almost totally different, and also that they do not derive from each other, because that was one of the questions. Some people imagined in the past that you could maybe, by some transformation, go from prismatic layer to the nacreous layer. In a sort of, if you make a partition of the prism, you can have some of Naker tablets, or if you put tablets together, not in a brick wall, but more in the columnar Naker, if you stick them together, they can form sort of prism. But actually, of course, this is not true. These two layers are very, although they are associated, they are not made of the same protein. So this work was published 2 years ago. And to summarize what we've done, about the high throughput screening techniques. First, we have an extremely diversified set of proteins. And I compared this to a sort of spear technique that we have the first rows, which are sort of prominent. Second row, maybe the little actors on the side, on the back side, hidden behind the curtain. But some quantitatively minor proteins may play key functions, especially enzymes. Well, we showed that even in associated calcified tissue, we had very different repertoires. And again, we plead for the fact that there is not a single model. We don't have our Escherichia coli of calcification. I do not believe in that. So each model is probably has its own specificities. Something that I didn't speak here, but which is very interesting, is because we also made a lot of comparison with Naker repertoires, and we saw that similar structure, like Naker, they can be produced by very different protein repertoires, which is sort of counterintuitive idea, but very appealing. Also, complex evolutionary history. Some of the proteins are probably fast evolving proteins, but we have also these old proteins from the deep past, like carbonic anhydrase, which are still in the picture. And this is here sort of it's a very rough model that I would like to work on. Imagine that here we have a sort of theoretical microstructure plan where each point is one matrix. It means that matrix can be associated according to the fact that they will produce this or Naker, prism, or cross-lamellar or something like that. And it seems that because of this variability of the proteins associated to one given microstructure, then what defines the microstructure must be something like an attractor, that you have the system which is pulled to produce either Naker or prism or cross-lamellar structure. Of course, I'm very careful when I say that. I'm a scientist, right? I'm not a scientist. No, I just want to make clear that there are people who could interpret this as in a wrong way. If you see what I mean. So, what next? Well, we are like this little character, and everybody will have recognized the little prince, Saint-Exupéry. We are just starting to cross the river. We don't know how deep the river is and how width it is. But it means that now we have to go on with the different aspects. First, we have to go to more quantitative and sequential proteomics to go more in detail to see what happens. And something that we have to work on is the supramolecular chemistry. And this is what is very needed in our discipline, that now we have the ingredients, but we don't know how they work together. So we need to go to have some supramolecular tools to go on with that. Continue to make comparative studies from model to model. Look at what are the shared themes, what are the lineage specificities. And I would like to mention the very nice work which has been published in the last years by the team of Bernie Dugnan in Australia, where they really show that a big part of the shell proteins have a very fast evolution rate. Some of them are very old partners, ante-Cambrian partners, but some of them were acquired much more recently. Another problem is the function of the matrix component, because so far we have sequences. Many of these sequences we cannot tell them, we cannot say, we cannot, even having a sequence, we cannot say what they do. That's the big problem. And I didn't speak about other partners in the story, what I call the post-translational modification, and also the non-protein components. And post-translational modification, we all know that they are very useful for calcification. In particular, phosphorylation of serines, but also glycosylations. And we have a lot of glycosylations in our systems. We should also mention small peptides, the lipids, and also metabolites. So, at the last picture, last slide. Here is the situation now in 2014. So, for peripherance, for silica system, few proteins and few sequence fragments for the calcifying sponges, for cnidaria, few tens of proteins more to come, arthropods, few tens of protein sequences, brachiopods. Now we work on brachiopods and we now also found very interesting things. Mollusca, as I said before, more than 3,000 proteins. Annelids, very few proteins. You know, they can make these calcified tubes. Echinoderms, few tens of sequences, and of course, chordates, a lot of sequences. So we still have to work on to fill this a little bit. And on this, I thank you for your attention. I just mentioned here these, let's say, partners or things who gave us some money. Thank you.